Now what is the second step in protein synthesis? That is initiation. Now initiation of what? Initiation of elongation. Now what happens in initiation is, what all do we have in initiation? First of all, we are going to have mRNA, and at that side this initiation would begin, where we have initiation sequence. That is AUG. Alright, one thing is this. Second thing is smaller subunit of ribosomes. Alright, if it is a eukaryote, then we are talking about 30S. If it is a prokaryote, then we are talking about 40S. Then what do we have? We have certain initiator proteins, but before that we have initiator tRNA complex. Then we have additional products like GTP, and that is guanosine triphosphate and magnesium ions. And apart from this, we have certain initiator proteins. We begin initiation with these 5 things. Now what do we have is, we have an mRNA, okay, it's like this. Now at this RNA, what happens is 30S unit, supposedly we are talking about the prokaryotic ribosomes, the 30S unit with an initiating protein comes and joins at the initiator sequence. Now at this initiator sequence, when this initiating complex 30S unit has come along with GTP and magnesium ions, these are initiating proteins. Alright, call it initiating unit. When this comes and joins with the mRNA, what happens is the initiator tRNA comes at this site. This is I am going to draw as we have. This is initiator tRNA that comes and joins like this. Initiator tRNA has amino acid methionine joined to it. Alright, the amino acid methionine is coded by, according to the genetic codon, that is AUG. Now AUG is the initiator sequence, as I told you. This AUG will be methionine in eukaryotes and formyl methionine in prokaryotes. So when this tRNA, that is either formyl meth or meth, that comes and joins with this 30S containing mRNA, 30S joined mRNA, what happens is larger subunit of ribosome comes over here. Okay, the 50S subunit also comes and joins at the mRNA ribbon. Now this methionine is located in the larger subunits P site. This larger subunit has 2 sites, one is P site and other is A site. You have to remember this, these are like rooms that are present. In one room comes and joins the tRNA, that is the space for tRNA, that is the initiator tRNA, that forms the P site known as peptidal site, and this is amino acyl site, right. Now this A site is meant for the next tRNA that would come and join over here, that would come and sit over here. So one is this, the second will come and sit in A site, and that would be according to the next coding sequence that would be present on mRNA. This part is mRNA. Alright, so we have P site, we have A site. The first initiator RNA comes, and tRNA of course, it comes at the P site, it reads AUG sequence. When it comes, the 50S also comes and joins. It is having 2 sites, and now the second site is ready to take up the next tRNA according to the sequence that is present on mRNA. I hope I am clear. I am just repeating it in a quick overview that what are the things that are required. First of all, you need an mRNA with an initiating sequence, that is AUG. From there we are going to start the process of initiation. When the specific proteins and tRNA, they read this sequence on mRNA, the 30S subunit of the ribosome comes over there along with the initiating unit, that is of course the initiator proteins. These initiator proteins make this 30S subunit join with AUG, okay, at that specific site, and at the same time what happens is an initiator tRNA complex which has methionine attached to it comes and joins at that particular location. As soon as methionine joins, this initiator unit gets separated, and what comes and joins is 50S subunit. 50S subunit has 2 sites, P site and A site, while the initiator tRNA sits in the P site. A site is empty and ready to receive the next tRNA, which would lead to the next step, that is elongation. In this process, apart from these things, that is one tRNA, mRNA, the subunits, there is another requirement, that is of GTP and magnesium ions. They enable the initiator proteins to come and let the 30S subunit join with the initiating sequence of AUG. They help in identifying. So this is all that you have to keep in mind when you read about initiation in the protein synthesis. The next step would be elongation.